Hey everybody, it's Mr. Chase bringing you live from my office the Microscope Parts Worksheet PowerPoint presentation. That's right, just for you. Okay, so you should have your 3-page worksheet out in front of you or with you, and I would strongly urge you to use a pencil to complete this worksheet because part of it you're going to be sort of guessing in the first section here where you have to label the microscope, and obviously you'll need to make corrections. The words you need are all listed here for that part. We'll go over it in just a minute. But everything's probably good if you write it in pencil, because then if you do make a mistake, you can correct it easily, and there won't be any fuss at all. Pencil and a working eraser, too. Okay, so we have this 3-page worksheet to go through, a lot of slides, a lot to cover, including our own microscopes and the teacher microscope. So let's get started. There we go. Fancy animation, waste of time. All right, microscope parts, everything you want us to know about microscopes, but we're afraid to ask. Here it is for you live from my office. So there's, you see some sample slides, images you might see looking through a microscope. You guys have, or we bought for you some slides here, some pre-done specimen slides, really cool. In fact, it looks like they're still, oh, I guess they're not sealed. But anyways, really fun, already done, already prepped for you and dyed so you can see them. Really fun stuff that you'll get to do later. But first, before you do any of that really fun, exciting stuff, you have to learn what a microscope is. What the names of the parts are, and how to use it. Very importantly, even with our little microscopes that are sort of designed for children, you can still damage and break them, and they're nearly $100 each to replace. So you don't want to have to do that. So please pay attention not only to what things are called, but how they work and how you should work them. All right, so we're going to spend quite a bit of time in this presentation going over what the different parts of the microscope are. There's a list of 13. You have it on your worksheet as well, all 13 of those things. You don't know what they are for the most part right now, but we're going to have you, in just a moment, use those 13 terms to label the diagram on your worksheet. So obviously, you mostly won't know, I don't think, what these are. If you do, awesome, good for you. This should be really easy, and that's super impressive, and you should let me know if you already know what these parts are, because that's amazing. But for the rest of us, we're going to be looking at these and taking our best guess and labeling it in pencil, again, lightly, what these parts are. So you've got 13 terms, 13 labels, and you've got 13 letters. You're going to just kind of write those by the letter, and let's see how you do. So at this point, you should pause this video. Take just a moment or two. Please do educated guessing. Don't just list them down in order. Don't be silly. Take your time and take some guesses and see if you can sort of get it so it sort of makes sense to you what these different parts are. After we finish this guessing, we're going to tell you exactly what the parts are. So if you haven't paused the video, please pause it right now. No, really, right now. Okay. Okay. Hopefully you're back now. And, okay, so you labeled them. We already did that. And here, I know you're really excited to find out, are the actual terms for these different parts of the slide. The microscope, sorry, on the slide. Let me see if I can get my little pen working again here. Okay, so let's start with A, obviously. Up here you have the, ooh, let's do laser pointer. That sounds really fun. Hang on. Laser pointer. There we go. Okay, that's awesome. All right, so starting with letter A, you have the ocular lens. Ocular lens, or eyepiece. Ocular meaning eye. So it's the part, obviously, that you look through. B is called the body tube. It's what the eyepiece or the ocular lens is connected to. Then going down to C, we're going to skip over that, come back to it. You have the arm of the microscope. That's clear from here at the top and all the way down to this other piece, which we'll talk about in a minute. So that's the arm, really important for safety. You'll see in just a moment. D, of course, is the base of the microscope. Makes perfect sense because it's the bottom, the base of it. Foundation. Going over to E over here, it's the light source or the illuminator if you're into Harry Potter. Okay, so that's where the light comes from. Our microscopes, just like this example here, have a light source. Some do not, and you'll see what happens then. F, going back up to the top, is the revolving or rotating nose piece right there. Okay, it's that round part that spins around. Going over to G, those are the objective lenses. Objective lenses, there's 3 of them typically. We'll talk about that when we talk about those lenses. H, where are you? H. over here, 2 knobs. It's a little tricky to see. Hopefully it's big enough on your screen. There's an outer ring, kind of a bigger knob, and then a smaller inner ring. The H, the bigger one, is called coarse adjustment knob. It moves things in bigger increments, larger increments. And then I, the inner one, moves the stage, we'll talk about that in a minute, in finer increments, much finer. So, and we'll talk about what you use that for. J, over here to stage, that's this flat piece. As you can tell, the lenses and the body tube and ocular lens, they don't move, they don't adjust. Well, the rotating nose piece does, which changes the objective lenses, but they don't go up and down. The only thing that adjusts for focus and to get things to where you can see them is this stage. So that's the part that's actually moving. On the stage, J, you get K. That's the stage clips. They're on top here. Makes perfect sense if you know this flat part is the stage, that the clip part on top is called stage clips. There you go. That's brought to you by Captain Obvious, by the way. JKL, condenser. This is this round piece underneath here. It focuses the light, brings it in, and kind of concentrates it, and it also can adjust the amount of light that's getting through with the diaphragm. So on the condenser is a diaphragm, and ours are a little bit different than this, and I'll show that to you when we get there. M is the diaphragm, and I believe that's all the parts. So if you have not corrected your guesses, please do that now so that your sheet has all these correct words correctly spelled and written neatly so I can read them. All right, so you got 13 terms. That's where they go. And if you haven't paused to stop and do that, pause the video now. Okay. Okay, welcome back. You should have corrected your guesses, all 13 of them. Hopefully you wrote them neatly and spelled everything correctly because it's just copying from your screen. You should all be able to do that. Just shows... Takes a little bit of effort and you'll be awesome. All right, so we're going to start at the top of the microscope and work our way down. We're going to go through what each piece is and what it does. All right, the ocular lens, the eyepiece, was the A in your diagram. All right, I'm going to highlight in purple for you. Ooh, magical. All right. So here's your fill-in now, so please make sure you have your worksheet handy, and then from now on you'll be filling in the blanks as we go along. So this contains a lens to blank the image of the specimen. What is it? I can hardly wait. Magnify. Ooh, exciting. Okay, so that's essentially what a microscope does, is it makes a very, very tiny thing or a piece of a tiny thing large enough for the human eye to see. So the first piece in that puzzle is the ocular lens or the eyepiece at the top there. The usual magnification of the eyepiece, because it, as you can see, it has glass in it kind of from the picture on the right, and that magnifies the image a little bit more. And the typical one is 10x. Okay, we either say 10x or 10 times, and that's what that eyepiece is doing. So every lens, or everything that has glass in it on the microscope over here is doing something to magnify the image, make a very small image larger so that you can see it. That's the whole purpose of a microscope. Okay. Now, some microscopes have, look at the picture. What do you think? Okay, where is it? Come on, there it is. 2 ocular lenses. We call them binocular microscopes. And in fact, I think I'll show you here in a minute. Okay, so you can write the number 2, by the way, or the word 2, whatever works for you. All right, so this is the teacher microscope that is our actual teacher microscope. See 2 pictures, one from the side here, and then one from, I guess, the front or the back. I don't know. Anyways, this would be how you look at it. So you put both eyes on this one, which helps obviously bring the image into focus. It's really fun if you get a chance to use this one. It's pretty awesome. But you do need to know how to use a microscope safely, because especially this one, which costs a few $100s, you can damage it and or break it if you don't use it correctly. If you're lucky enough to have your teacher let you use it, then you'll have to demonstrate that you know how to use it safely. Also, you can kind of see here the little USB cable coming off the back. It's got a camera here in the tube, which allows the image to also be visible through the eyepieces here, the binocular eyepieces, or on a camera on the screen of the computer. So this microscope's pretty fancy and pretty awesome. And also here we'll talk about these coarse and fine adjustment knobs, but here you see a great example. It's even got indicators on it to show you kind of how precise you can adjust it. So, and you'll see more about that later. Got a cool light source here, nice and bright, and again you can adjust the aperture to let in as much as you need to. And you guys won't touch this without permission, though, sad for you, but it is really awesome. Okay, so talking about parts of the compound light microscope. Here we go. So we're moving on from the eyepiece at the top here to B, which is the body tube. And I believe you have more. There's the purple highlight. Very exciting. Okay, it blank the eyepiece to the objective lenses, and that is connects. It connects the eyepiece, this top part that magnifies about 10 times, to the objective lenses down here, typically 3 of them, and through this capsule right here. It ensures the correct blank of the microscope components, and that is alignment. It ensures the correct alignment of the microscope components, so it keeps everything working as a nice straight line. Obviously, if something gets out of alignment, the microscope won't work properly, and you can't see anything, or what you see will be a very poor image of what you're looking at, so we don't want that to happen. And it ensures the correct alignment of the microscope components to correctly, here we go, direct the light from the specimen, that will be down here on the stage on a slide, into the viewer's eyes. That's you. You're the viewer. Okay, here we go. Watch. Ooh, that was exciting. Okay, the light goes through the bottom of the microscope, through the stage, and through the lenses, through the eyepiece, and then into your eye. So it's traveling through several stages, but as it's doing that, it's getting bigger and easier to see, and hopefully more focused for you. Now, C, the arm of the microscope. Okay, it starts up here, right next to this capsule, goes all the way down to the bottom of the microscope where it connects to that flat part called the base, which we'll talk about in a minute. The arm of the microscope is probably one of the most important parts of the microscope in terms of handling it and moving it and doing so safely. Which you'll see. Ooh, highlighted in purple. Did you miss it? That's over there on the left. Okay, it blank the body tube to the base, and that would be connects the body tube to the base over here. One blank should be around the arm when blanking the microscope. Anybody? Okay, one hand should be around the arm, obviously right here, when carrying the microscope. Okay, I don't know that you guys will be moving the microscopes, but some of you might be helping bring them out or put them away or both. And you must be careful with these microscopes. Again, the student ones are not terribly expensive, but I don't know if you want to be dropping $100 to replace them if you break them. So please be careful. And plus, you just don't want to break it because then we don't have enough for everybody. All right, the other hand, you have one carrying it here in the arm. That should be nice and solid. The other one should be under the base. Okay, so it's kind of like this. I have our microscopes right here in a bag. Hopefully you can see it. So you have one hand on the arm here, and then one on the base. And that's how, let's see, out here. That's how you'll carry it. Okay, and there's a picture for you. See, one hand on the arm, one hand out of the base. Awesome. This person's doing a great job, and you'll do a great job if you have to help. All right, there's the base. We're working our way down the microscope. Ooh, purple. Very exciting. Okay, it blank the weight of the microscope it supports. Okay, obviously it's going to rest on the table or desk wherever you're working with a microscope, and it has to be solid enough and stable, because the last thing you need with from a microscope is for it to be wobbly, right? That would not be helpful. So they tend to be a little bit heavier to help it rest steady. If it is moving, if your surface that you're working on is moving, it's going to be really hard to see because the slide itself will start jiggling, and you'll see once you experiment with these a little bit or get to use them that the focus is really hard to get, and once you get it, you don't want to lose it by it vibrating or moving around. All right, the base also contains the blank and the blank. What is that? Electronics and the light source, okay? On our student microscopes, they do have a built-in light source here. I'm going to go ahead and take the bag off. It's there to keep what? Anybody? Keep the dust out. Notice how the eyepiece has a protector, and so does the stage, to keep dust off these components. Because one of the enemies of microscopes is dust. All right, so we have a light source right here. Ours are battery operated, so it's not quite as fancy as the electric ones. And some of the microscopes, the adjustments are going to be electric adjustments. There's like low motors, but ours aren't. Again, the student ones are not that fancy. In fact, if you look here on our actual microscopes, you'll notice there's just one knob. So we actually don't have a fine and coarse adjustment knob, but they are on the teacher one if you remember back. Oh, and ours have the adjustment on both sides. That's kind of nice. Okay, one hand should be blank the base while blanking the microscope. Ready? I think you got this one. Under the base while, again, carrying the microscope. Okay? Pretty simple, but it's super important. Simple, but important. The other hand should be holding the arm. We've already talked about that, and you should know that. And there's another picture in case you missed it the first time. I know it's super exciting. Okay, light source, kind of working our way back up again. You see it here. They have a close-up for you. It's going to highlight in purple. I know, it's so exciting how that happens. Okay, it sends the light blank through the blank. Upwards, there's a shocker, sends it upwards through the, there, see the little arrow in case you weren't sure what upward was, through the condenser lens. Woo, condenser lens. Okay, so the condenser lens is right here. So you see it kind of, again, bringing that light in, focusing it, because you want to have the light as focused and bright as possible and not just going out into the room, because that would be useless. So they have this lens on there, the condenser lens, to try to focus the light and maximize the benefit of the light. And through the blank in the stage. So it's going to be the hole. Fancy scientific word hole. Okay. Oh, sorry. Condenser lens is right here. Okay, it's this piece. This is the light source, then you have your condenser lens, and then that's the hole right there on the top. And there'll be a specimen on a slide. The slide's the glass part. The specimen is what's on the slide. Older microscopes used to use, yes, and I did use ones like this when I was in school, or at least see them. Okay, they used a mirror. That's right, folks. We didn't have little lights. We had mirrors to focus the light from the room through the hole, the aperture in the stage, so that's how we lit up our stuff. That was the only thing that they had at that time. Okay, so you may see these. You may, I doubt it, but they have mirrors and they reflect the ambient light upwards. Okay, so older microscopes used mirrors to reflect the ambient light upwards, and there you see here in the example, the light's coming in, bouncing perfectly up the mirror and into the hole in the stage. Okay. So these are the revolving or rotating nose piece. That's this round part technically. Okay, revolving or rotating nose piece. Right there. The blank are attached to it. That would be the objective lenses. That's these three lenses. Our microscopes do have three objective lenses. As you can see, they've got little numbers on them telling you how much they multiply. There's 4x. I think that's 10x. Yeah, 10x, and then that's 40x. Okay, so 4 times, 10 times, and 40 times is the amount of magnification. The blank the nose piece allows you to blank between different lenses. Rotating, that's turning it. The nose piece, that's this circle, again it spins to switch between different lenses. Pretty straightforward. Okay, now we're moving down from the rotating nose piece to the objective lenses. There's 3 of them here. These lenses further blank the image of the specimen. They've further magnified, as you might imagine. And you have a low, medium, and high magnification. They have the word scanning here with the low one because that's what you should always start on when you're looking at a specimen, to get the image centered and get focus as best you can with that minimal magnification. So it's called scanning because you're just trying to kind of get your specimen, get the slide specimen located appropriately, and then you can move on to those other magnifications to try to get a better look at what you're looking at. The magnifications are usually blank, blank, and blank, so those are numbers. And we go 4x, 10x, and 40x. So our student microscopes, I think I might have said microphone somewhere in there, but the microscope, the objective lenses are exactly like average. The teacher one, though, is not. Okay, there are usually 3 lenses, as you might imagine. Every picture we've had has the 3 lenses. I think our teacher one, if you go back and look, pretty sure it has more than 3. I think it actually has 4 of those objective lenses. Okay, but some have, and I just said that, 4, and you can see that there, that they have the 4 lenses. Okay, this rotating nose ring has 4. As the power increases, the magnification becomes obviously, there you go, see kind of an image that starts out small, you start changing the objective lenses, and then you can see closer and closer, so the magnification becomes larger. The magnification becomes larger. The image doesn't actually get larger, it just looks larger. Okay, but the field of view, that is how much of the slide you can see, or the specimen, it actually gets smaller. All right, so as you get closer, the magnification is larger, but the field of view, that is what you can actually see, actually becomes smaller, is what you might imagine, because you're zooming in more and more. All right, so we're going to talk about these coarse and fine adjustment knobs. Again, your student microscopes don't have them, but some do, and like the teacher one does. And as you move forward in science, hopefully, and high school and beyond, you'll likely run into microscopes that have these coarse and fine adjustment knobs. Okay, so the coarse knob is bigger, and it's also the outer one. And let's see, the first knob you should use, and always under low power. Never use it in high power. Okay? Can anyone guess why you wouldn't want to use the coarse knob, the coarse adjustment, in high power? Anyone? Anyone? Okay, so the reason is because if you have this big lens on, which is your high power, and you're moving it a lot, it is entirely likely, if you're not careful, which it's hard to be, you actually smash into your specimen and either break the specimen slide, break your lens, or both, and damage the microscope. Okay, so it moves it too much. And even on ours, I noticed, okay, if we have this high powered one on, if you kind of look in there, if you can see the space, and like if you imagine that there was a slide there and I move this up, see how that's like actually going to smash the slide into the lens? That's not good. That's going to be bad. One of the things they don't say in here in terms of use, and probably Mrs. Jones will tell you, is that if you are adjusting, since ours only have one knob, you're going to adjust with the high-powered lens, the 40x lens. You'll want to make sure that you do adjust it. Start at the top, start with the stage at the top, and work down. That way, you know it's not going to smash into the lens. If you do the opposite and just kind of start adjusting it, it's very likely you'll smash the stage with the specimen slide right into the objective lens. And again, you can break the slide, you can ruin the slide, you can break the lens, you can break the microscope. So that's why it's saying never use this outer ring, the coarse one, which moves the stage a lot. Okay, we don't have the inner and outer one, so you're just going to have to be really careful. Start at the top, start with the stage at the top, and then move it down. Hopefully you've gotten into focus with the other powers lenses first, and that should help you a lot, kind of have the stage in the right place, the stage with the specimen. Okay, so the fine adjustment knob, and again, this is going to move the stage in way smaller increments. It's the second knob or the inner knob, and that's the one you should be using under high power. But even if your microscope does have these coarse and fine adjustment, you still want to be careful to make sure you're starting with the stage up and move it down only, and not start it up and move it up right into the lens, because you can still smash it. So regardless of which one you're using, you still have to be careful not to smash them. Okay, and you're going to use it for exact focusing. And you'll see how tricky this is once you guys get to experiment with these microscopes, which is super fun, by the way, with the prepared slides that we have for you. Both knobs move we already said this, the stage up and down to help put the specimen in, and we kind of talked about this, to get it in better focus. When you're working on a really small item and it's very thin, the focus has to be pretty precise. If it's a thicker object, you have a little bit more to play with, but the thinner the specimen is, the more exact your focus has to be. Otherwise, it goes out of focus and you see nothing but blurry nothingness. And sometimes it's hard to tell that there's even anything there. All right, some microscopes have two knobs. You see here this one has two. It's got the bigger knob and the smaller knob, which still should be a clue to you which one does what. Okay, and they would be located one on top of the other. Okay, H and I, and you see the split here, that the larger knob here, the outer knob is still going to be the larger, do the same function as the larger knob on the microscope with two knobs, and then you see the smaller knob is also a smaller knob here, and it's going to do the same job as the the one that's inside on the one that's together. Okay, so the smaller one on the bottom is always the fine adjustment top, and the big one's usually the coarse or the larger movements. Okay, so the stage, we've talked about it a little bit. So we're just going to identify the stage here. There's your purple highlight, very exciting. The stage is where you place the blank, which contains the blank. And we've already said this too. It contains, oh, look, it's highlighting it over there in blue for you. Okay, it's the slide, which contains the specimen. All right, again, make sure you're spelling these correctly. Take your time, pause the video if you need to, but spell the stuff correctly and write it neatly, please, so that we can read it. the stage, contains a blank that allows blank to pass through the stage and onto the specimen. Okay, you probably could guess this now. It contains a hole, which is basically nothing. Okay, open space, a hole that allows light to pass through the stage and on to and through the specimen. Okay, and the stage clips, again, it makes sense. They're on top of the stage. They help clip the slide in. And just a little tip for you on these: you don't you don't try to jam your slide under the clips. What you want to do is rotate the clips out of the way, put your slide where you think you want it, again under the low power to start with, and then carefully rotate those clips. You kind of have to lift them a little bit. You see they're kind of spring-loaded there, down onto the specimen slide on the stage. And you can see ours are exactly like that. They're kind of spring-loaded, so they have some tension down, so you have to lift them up, but you move them out of the way first, get your slide position, then lift them and rotate carefully and hold the slide down with your specimen. Okay, again, don't ever try to jam your slide under. That's likely to break the slide and or scratch the stage and or ruin your specimen. Okay, so you have to be careful with these. They're not indestructible. All right, again, pointing out with yellow now where the clips are on your stage. Okay, the stage clips blank the slide onto the stage. You probably guessed this. They secure or hold the slide onto the stage because, again, working with focus, you don't want your specimen sliding around or moving, because if you get the focus where you want it and then the slide moves, then you've lost focus again. So you have to kind of constantly repeat. So the stage clips are important, just like every other piece part on the microscopes are. All right, condenser lens. This is underneath the stage, underneath the hole, and it's what's going to focus your light, kind of adjust the amounts like an aperture. And you can see here this one. Let's see if I can show you over here. Okay, so this is called the condenser lens in there. And okay, so let's go through this and I'll point out what this little guy does here, the little lever. The lens under the stage that blank focuses light from the illuminator, there's that word again, through to the hole in the stage. Okay, so it's going to take this light, focus it in. You can adjust the amount, and then it's going to send it through the hole in the stage, through the specimen on the slide, and then up through your objective lens and so on until it reaches your eyepiece. Okay, the diaphragm, and I think it's going to highlight over here. Yeah, the green part. Okay, so the diaphragm contains a dial that rotates to blank the blank that reaches the specimen, and that is to adjust the amount of light that reaches the specimen. Our student microscopes, I hope you guys can see this, they have a dial, but our aperture, okay, the diaphragm is a little different. I don't know if I can even show you, but it doesn't adjust the hole. It just spins and has holes of different sizes to allow different amounts of light through. So it's a little different, but it works essentially the same. Okay, so there's your student microscope. I included it. You'll notice on our student microscope, we have the 3 objective lenses, eyepiece, and so on, the body tube. We've got the arm of the microscope, base, and all those things. And we do only have the one adjustment knob, not coarse and fine, so that's a little bit of a challenge. You got to be careful. Our light source down here, and then kind of a cool thing is we also have a light source on top. Some slides and some objects, like for example this flower, you're not going to really be able to see through very well because it's kind of thick relative to something that's on a microscope. So in order to see that, you'd really want the light coming from the top because the light's not going to penetrate through that object. So there are some things, sometimes, occasions where you'd want the light to come from the top, and that switch is on our microscope back here. There's a 1 and a 2. Okay, and so 1, oh, what is that? All right, so the number 2 sends the light from the top of the microscope to the stage, and then if it turns it off in the center, and then on the other end, it's going to send the light up through the light source from the bottom, just like typical. So that's kind of a cool feature of our student microscopes. All right, so I think that's everything for your worksheets. You should have that all filled out now. If you missed something or need to revisit it, please do so. You do, once that 3-page worksheet is complete, neatly complete with your name on it, if it's not already, it should be. You've got this other page that's going to, again, you're going to practice labeling and identifying on a sample microscope all these same exact 13 different things, and you're going to color code these little squares to match the color that you color in neatly on your microscope. Okay, the challenge will be, as I sort of mentioned to you already, the coarse and fine adjustment knob. I guess what we can do is just maybe draw a little arrow or do an outer color and an inner color here. We're going to pretend that this is adjusting it. I guess they were maybe thinking that this one had an upper and a lower knob, but let's just go with this one down here and do an inner and outer one. Okay, so we got 13. You're going to color code this one. You are going to turn this one in. Make sure your name's on it, of course. And then after you do this one, you're going to, oh, that's not the end. Okay, so after you do this one, there's a paper that I didn't put in here, but it's got just the lines. And that one I encourage you to use to study and to practice labeling which part is what. And what you might want to do is take that one with just the lines and maybe cover it in like a sheet protector or something like that. And then you could write on it in a grease pencil or some sort of like a whiteboard marker, and then you could just keep erasing it and keep practicing. That would be a great way, because you do need to know these 13 parts, and you need to know them well for the test. And so you will have practiced it a minimum of 3 times. If you just want to use pencil and kind of erase, I don't know if that's going to work very well unless you write really lightly on that other piece. Okay, so your 3-page thing you're turning in that we just finished. This page you're going to turn in. We're going to give right back to you as quick as we can so that you can use it to study. And then you've got the other blank one with lines so that you can study and practice with that one. So lots of practice, because you need to know all these parts and really comfortable. And for most of you, this is going to be brand new stuff. So you need as much practice as you can get. All right, if you have any questions, please let me know. Otherwise, good luck and be good for Miss Patty, or else. Bye.